This Week in Virology, the podcast about viruses, the kind that make you sick. It's time for This Week in Virology. This is a special episode recorded on November 7th, 2014. Hey everybody, I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. I'm coming to you today from Manhattan, not New York City, which is where I usually am, but Manhattan, Kansas, and I'm at Kansas State University. I'm the guest of the Division of Biology at this university. And I have to say, I like Kansas already because on the way from the airport last night, I could drive 80 miles an hour without any problems. The speed limits is 70, 75 miles an hour. And that's really cool, and the roads are pretty straight. So not something you can do in New York. So I have two guests today from the Division of Biology. They're both virologists, as you might guess, and we're going to talk about the work that they do here. And on my left is a professor in the Division of Biology, Rollie Clem. Welcome to TWiV, Rollie.
Hi, thank you. Thanks for having us.
And on Rollie's left, also a professor here in the Division of Biology, Lorena Passarelli.
Thank you for having me here.
Welcome, both of you. Thanks for coming. And we have a nice, enthusiastic audience, right?
Yes.
Who listens to TWiV? Almost everybody. And afterwards, after you hear this, everybody will, because this is just a cool way to communicate science. So I want to start by asking both of you to tell me where you were raised and educated and how you got here. Let's start with you, Rollie. Where are you from?
I grew up in northern Minnesota. My father worked for the Department of Natural Resources, so I grew up on wildlife refuges in northern Minnesota. I went to college there at Moorhead State University in Moorhead, Minnesota. Mm-hmm. And then eventually went to Georgia, University of Georgia for graduate school. Where Kathy Spindler was one of my...
I just visited Athens a month ago.
Yeah. She was one of my committee members there, so I know her. And then did postdoc at Johns Hopkins before coming here.
So your dad's involvement in outdoor work got you interested in science, you think?
I definitely, yeah, I was always interested in biology and started out thinking about wildlife biology as a career, but then in college when I took microbiology class, that changed everything. Got interested in small things.
And then you decided, while you were still in college, to get a PhD in
initially I started with a master's program, but then I transferred to a PhD before I finished the master's program.
So my episode at University of Georgia will be out next Sunday. So I spoke with two virologists, Biao He and Zhang Fu.
Okay.
I don't know if either was there when you were there.
No, I don't know. Not that I know of.
But anyway, it was a lot of fun. You should check it out. But the audience wasn't as good as this one.
Naturally.
And you guys have the edge because I couldn't find anything to eat after 9 p.m. in Athens. And here last night I had no problem. So yeah, Manhattan is winning out over that. So you went to grad school at Georgia, right? What did you work on there?
Baculoviruses. So I was in Lois Miller's laboratory there in Georgia.
So that's where the baculovirus interest began, way back as a PhD student.
Well, actually it began when I was an undergraduate. When I had to do a senior seminar as an undergraduate, I had to pick a topic to research, and I just happened to come across a review article about baculoviruses and how they could be used as biological pesticides, and I just thought that was the coolest thing ever. And then I worked on plant viruses for a couple of years, but then eventually got into baculoviruses.
So your postdoc, you said, was at Hopkins?
Yeah. With
who?
With Marie Hardwick and Diane Griffin.
So you worked on RNA viruses, right?
Yeah, I was mainly working on apoptosis, actually, in mammalian cells, but we did some work with Synvis virus, RNA virus, while I was there.
And then after that, did you come right here to Kansas for your first faculty position, right? And you started a lab to work initially on baculo, or was it also other arboviruses?
No, I was working on baculoviruses for about the first 10 years, and then transitioned some into arboviruses. Still doing some baculovirus work, but mostly arbovirus work now.
So you've been here how long now?
Since '97. So.
Must like it, right?
Yeah, it's been a good place. It's a nice town to live in. There's a lot of good support here, so it's been good.
And plenty of viruses.
There's always plenty of viruses, right?
Well, we're going to talk about what you work on in a minute. Lorena, where are you from?
I come from a little bit farther away than he does. I usually make people guess where I'm from.
Want me to guess?
You want to guess? Since my accent has evolved through the years.
Well, my first guess would be Argentina. And then if that's wrong, I would say Italy.
Last name is Italian. Grandparents were Italian. I
Yeah.
come from Guatemala.
Guatemala.
Yes.
All right, I was not too far with Argentina, right?
Almost a continent away.
So Passerelli
Passerelli's
is
Italian.
very similar to Racaniello.
Yes.
Lots of
Yes.
vowels and an I at the end, or
And
no?
double letters, yes.
So your parents were from Italy?
My grandparents were from Italy.
And someone moved to Guatemala at some point?
My grandfather moved to Guatemala. They started a business there, and I grew up in Guatemala. And I came to the United States, 36 years ago, believe it or not.
How can it be? You're only 25.
Yeah, well, 26.
I'm sorry.
So,
What did you come to the U.S. to do, to go to school?
to go to school. All my schooling was here.
University?
Yeah, I went to, in Boston, I went to a private school called Simmons College in the Longwood area where Harvard Med School is, and Beth Israel, Brigham's Women's Hospital, all that little center
Right.
in Boston. From there, I moved to the University of Connecticut in Storrs, where I got a master's degree in microbiology. I did work with bacteria, and then I went to the University of Georgia.
For
I got my
for
PhD.
a PhD?
And I went to Lois Miller's lab in the in the genetics department, and I studied baculoviruses there.
That's where you met him, right?
Yeah, I met him there, yeah.
Okay. And then where was your postdoc?
And then I postdoc at the NIH in Bernie Moss's lab working with pox viruses.
Okay, so
Yes.
yeah, you didn't.
Yes.
You're too young to know her, right? Geelong.
Yeah.
That's the same lab, right?
Yes.
That's
same
why he's
lab,
here, I
yeah,
guess.
the same lab, yeah.
You worked on pox viruses,
I
right?
worked on pox viruses, gene regulation in pox viruses.
And that's when you were at Hopkins, right? So that
Yes.
worked out, right? So
Yeah.
you were
You lived
kinda
in the
close.
middle.
We
Yeah.
lived in the middle.
Yeah.
Between Baltimore and Washington, D.C.
Okay. And then you both came here after that for your first faculty position. You've been here ever since, both of you, right?
Yes, we have. They treated us well. We've stayed.
So this is an interesting question. You know, it's hard to get one faculty position. So I met my wife in graduate school, but she's a scientist, but she's in a different field. And we always thought for 2 career science jobs, big cities are good. But you obviously have found it to work in a small city or a small town, I guess. And I wonder if you could just give us some insight. Today, if a couple were looking for jobs in science, would it be harder than when you were looking, do you think?
I think it's more difficult to create new positions now.
Yeah.
I think people are more aware of it, and I think it's more common to have couples in the same career,
Yeah.
which might make it easier. We had people, one person retiring and another position opening at the same time, so there were 2 positions when
Yeah,
we came
yeah.
here, so we lucked out. So I've always been asked whether people should reveal that they're together when they apply or not. And that is a tricky question, and I'm not sure what's the best strategy.
Yeah, I mean, I would always vote for openness because,
yeah.
as Mark Twain said, if you don't lie, you don't have to remember anything, right?
Yeah.
But,
well, I mean, keep in mind that it's not the job of the search committee to decide whether there's 1 or 2 positions. They have to
Yeah.
interview the best candidate, period.
Sure. That's why I would think now, with even getting one position hard in science, that a city would offer more opportunities. So in New
Yes.
York, when we are interviewing someone and they say, I have a partner who's a scientist. There are many other universities that we could go to and say, do you have positions for someone else, you know? Whereas here, I guess you just have one place, so
Yeah.
it could be a little harder, which is, I mean, it's not unusual to have couples in the same field. I mean, in fact, you're the second we've had on TWiV. We had Jean-Michel Claverie and Chantal Abergel from Marseille, who work on giant viruses.
Yes.
They work together.
I've met him.
And they have gone all over the world in their PhDs and postdocs and so forth, and it works. So my feeling is if you want to do it, you just have to try. What do you think, Rollie?
Yeah, I mean, it's difficult, definitely, but there is more awareness of it as being an issue, I think, now. But at a place here like K-State, like you said, it's harder to find the resources to do that.
So maybe if you were doing this today, just looking for a job, you wouldn't end up here. You'd end up in a bigger city.
Yeah.
Well, I think you apply everywhere, right? And you just see where you end up. But
Yeah.
that's
Do you view yourself as role models for this at all?
I wouldn't say role models, but I think I've had some postdoctoral fellows asking me about what to do and how to go about it. We have here three couples in the Division of Biology.
Okay. Well, it makes sense in a way because it being the only place in town, right?
Yeah.
You would attract couples. Okay. Well, I thought that would be an interesting thing just to explore since we don't encounter it very often.
My only advice would be if they get a position in the same place, they need to keep independent work. I think each person needs to develop their own program independently.
Yeah. So each of you have separate
We
laboratories,
had separate labs,
right?
and we never collaborated till we were professors,
Till
full
recently,
professors.
Why is that important?
I think you need to show that, well, when you are non-tenured in the tenure track position, I think you need to show some independence.
Yeah.
And I think when you're a couple, that needs to be further emphasized.
Yeah. I think that's good advice because we always tell young faculty, publish on your own.
Yes.
Otherwise, the tenure committee is going to say, oh, you have all these other authors. We don't know what they did and what you did.
And especially if you're both at the same stage, then, and you have common publications, then who did what becomes a more blurry line, especially if you're coming up at the same time. So I think it's good to have independent work.
Okay. So when you go home, do you talk about science at all?
I always try to avoid it. She always
I always try to
brings
talk
it up.
about it.
Our kids are always like, please, let's not talk about science
Really?
at the dinner table. But yeah, inevitably it does come up. We do end up discussing a lot of things at home, which I think is good and bad. It's good in a lot of ways. More good than bad, I think.
Well, you know, we have 3 kids, and I always try to talk about science, but my wife doesn't want me to. She's not a virologist. And she always says, Vinny, turn down the viruses, please. She doesn't want me to talk about it. But my kids love it. So when she's away, I talk to them a lot about viruses. But what I find interesting is that despite having a culture of science in our house, my kids don't want to be scientists, at least not life scientists. They want to do my 2 sons want to be computer scientists. So they're very aware of science and the importance of it in vaccination and all this, but they still want to do other things, which I think is neat.
We did try to infuse a little bit of science. I mean, we get Scientific American, and we talk science. A funny story about that. My daughter, when she was maybe 7th grade or so, the teacher told them, I am writing a grant for an educational program that she was envisioning. And she asked the class, does anybody know what a grant is? And my daughter raises her hand and she says, it's something that is very stressful and it takes a lot of time.
That's great. That's good.
So she was the only one aware about grants.
Yeah, my kids bring home science stories. My son told me once they were talking about vaccines. I think it was in 9th grade. And they're talking about the HPV vaccine, papillomavirus, which is a hot point among parents, right? Because many parents feel that if their kids get this vaccine, it's going to make them more promiscuous, right? As if anything would stop them one way or the other, right? But So one girl raised her hand and said, my father said we can't have the HPV vaccine because it gives you polio. And my son said, my dad works on polio, and that's not true. and she said, well, I believe my dad. So it's very interesting what kids bring from home. Anyway, I want to talk about your science. We've never had a TWiV on baculoviruses, so I'd like to talk about that quite a bit. And I also want to talk about viruses that are carried by mosquitoes, which are things that both of you work on. So let's start with mosquitoes. There are lots of different kinds of mosquitoes in the world, but not all of them carry virus infections. Is that correct?
Actually, very few of them do, actually.
So do you know roughly how many different kinds of mosquitoes there are?
What I've read is there are over 3,000 species recognized.
Okay.
And probably something like 50 or 60 of those species can carry different types of viruses.
So what are some of those species that carry viruses?
Probably the most important one is Aedes aegypti, the one that we work on, which is the yellow fever mosquito. Carries dengue, chikungunya, yellow fever. Culex mosquitoes carry West Nile. Aedes albopictus is another Aedes species that carries chikungunya and dengue.
Any others? So those are the ones I'm aware of too.
Those are the major ones. So, I mean, there are a lot of other Culex and Aedes species and things like that.
But so the Culex is the genus, right?
Yeah.
What other genera would be bearing viruses?
I can't think of any other ones right now, but there are a few other ones.
Okay. But mostly the
those
main
are the
medically relevant ones are the ones you've mentioned. And we know this because people have trapped mosquitoes and find viruses in them, correct?
Yeah.
So do we understand why it's limited to these genera?
That's not very well. I mean, that's mainly one of the things that we are interested in is what determines, that interaction between the mosquito and the virus, because it is very specific. So among the viruses, usually they're only carried by a small number of, maybe only one species or a small number of species. So in most cases, they cannot, mosquitoes are resistant to being infected by the viruses for one reason or another, and so that's a big area of investigation. So
let's say in a population of Aedes aegypti somewhere, if you sampled the population, what fraction would be carrying viruses?
It's pretty small, actually. Even when there's an active epidemic going on, it's, from what I've read, less than 10% of the mosquitoes are actually carrying the virus.
Yeah. Okay. So that's, I've been thinking a lot about Ebola lately, and it's a similar situation in bats. About 1% of bats seem to be seropositive for Ebola at any given time. But that can cause a lot of trouble, obviously. Right. So in order to transmit a virus infection among mammals, say, does the virus have to replicate in the mosquito always?
Yeah, at least with arboviruses and mosquitoes. Yeah, there are some plant viruses that can be transferred passively, but most of the time it requires active replication in the vector. So
all the viruses you mentioned, dengue, chick, West Nile, your Sinbis, right? Those are the main ones, correct? All of the yellow fever, right? All those replicate in the mosquito.
Right.
Do they replicate it in different places in the mosquito?
They have to. I mean, initially they replicate in the midgut cells, which are when the mosquito takes a blood meal, the virus is in the blood, so it has to initially replicate in the gut and then escape out of the gut. And the tropisms are slightly different for different viruses. Most of them replicate in the majority of the tissues in the mosquito. But the most important one is the salivary glands. They have to replicate in the salivary glands to be transmitted to the next host when the mosquito takes another blood meal.
So can we go through what happens? A mosquito lands on me and this is a female mosquito, right?
Only females, yeah.
Only females, and takes a blood meal. So inserts that proboscis, is that what it's called?
Proboscis.
Proboscis. Yeah. And I have to be viremic in order for virus to be transferred to the mosquito, correct?
Right.
What levels of virus in the blood are needed, do we know?
It has to be fairly high. That's one of the reasons why humans are dead-end hosts a lot of times, because they don't get high enough viremia levels to transmit from one human to another. So it depends on the virus. With West Nile, most of the transmission is coming from birds to humans, but not so much from human to human.
Because the birds have high levels of viremia.
Higher levels, yeah.
Okay. And so all the human viruses that go from person to person via mosquito, those reach high levels in the blood. Do we mean 10 to the 5, 10 to the 6 viruses per mil or higher?
You're testing the limits of my knowledge. But
me too.
Yeah, I don't know for sure what the levels are. From my impression is 10 to the 7, something in those range. But so dengue is one that mainly is only a human virus and is transmitted between humans by the mosquito.
Right.
Doesn't really have animals.
There's no animal in between, like West Nile, right?
At least not with the urban dengue.
And what about yellow fever?
Same thing. There's urban yellow fever, which is human to human. There's also jungle yellow fever, which is more monkeys are involved with.
And chikungunya is the same?
It's also human to human, as far as I know.
And Sinbis?
Sinbis is also a bird virus, so it infects birds as well as people.
All right, so the mosquito is taking a bite. If you're viremic, the virus is ingested and it goes to the midgut.
Right.
So is there a foregut, a midgut, and a hindgut, I presume?
Yep.
Passes through the foregut. Is the midgut analogous to our intestinal tract?
Yeah. So the midgut is the majority of the gut in the mosquito.
So in order for this to be transmitted to another animal, the virus has to get into the circulation of the mosquito. Is that correct?
Well, mosquitoes don't really have a circulation system. They have an open circulatory system. So, but the virus does have to get through out of the midgut across the basal lamina that underlies the midgut cells and into what's called the hemocoel, which is kind of the open body cavity in the mosquito. And then it's going to circulate around in the hemolymph, which is the equivalent of the blood, as that gets circulated around inside that open cavity. But eventually it ends up infecting most of the organs in the mosquito.
And the key one is the salivary gland.
Right.
Because then when the mosquito bites the next host, it delivers virus along with, well, it's going to take a blood meal. So how does the virus, when the mosquito is pulling blood out, how does virus get delivered?
As the mosquito is taking blood, they're also injecting saliva into the host, and that's important actually for the mosquito to be able to take a blood meal effectively, but also suppresses immunity in that immediate area of the bite, and does probably a lot of other things that we don't understand very well.
So just before taking blood, it injects some saliva?
I think it's as it's feeding, it's injecting saliva.
So it can do both?
It can do both, yeah. And so that's when the virus has to replicate in the salivary glands and be shed into the saliva to be injected.
Okay, so if the virus doesn't get out of the midgut, it will be shed it will be excreted by the mosquito, I presume, or degraded on the way out, right?
Well, any virus that doesn't attach, would be excreted out.
So there are probably a number of viruses that mosquitoes take up, but they don't replicate and get shed, right?
Yeah, exactly, and that's one of the main thing that we're studying now together as a collaboration, is that what allows the virus to, if it infects those cells in the midgut, how does it get out of the midgut? So there's a barrier to infection in some cases. So sometimes they can't attach to the midgut cells and even infect them. Sometimes they infect them, but they don't spread cell to cell. Sometimes they can do that, but they can't escape out. So there are several different barriers that the virus has to overcome.
So in humans, is the layer of cells sort of similar to our intestinal tract?
Yeah, it is.
Layer of epithelial cells, a basement membrane below it.
Yeah.
And we have these M cells too. Do mosquitoes have those?
I'm not sure what M cells are.
These are areas of the intestine that are very thin, and they're used to sample the antigen content of the intestine. So cells, antigen-presenting cells can reach through, dendritic cells can reach through and grab antigens. And they're thought that that's how some viruses get through the human intestine.
I'm not aware of something like that.
Yeah, probably if you don't know about it, then mosquitoes must not have them as well. Because that's a way that viruses get into humans. But also, replication, in humans anyway, replication in the epithelium causes inflammation, which can disturb the basement membrane and allow viruses to pass through. I don't know if that also happens in mosquitoes.
Not that we know of.
Different
I mean,
things,
yeah.
right? So there are multiple blocks to viruses getting across, and that's one of the things that you study. You
Yeah.
say. So are the receptors that viruses attach to the same as in other cells, mammalian cells, for example?
Receptors are not that well known with arboviruses, at least not in the mosquitoes. There have been some putative receptors that have been identified for some of these viruses, but the ones that are important in the midgut are not very well understood.
Okay.
But the virus does have to attach. There are the surface proteins of these viruses. There are mutations in those proteins that can eliminate or reduce attachment, so they are attaching to something.
So as you might imagine, we've been getting a lot of questions about Ebola virus lately, and one of them that I think we addressed is whether the virus can be spread by mosquitoes or some other arthropod vector. And I was wondering if you had any thoughts about that.
I think it's unlikely, just because in almost all cases the viruses do have to set up this replication in the mosquito and spread in the mosquito to be. So they're not simply just flying syringes that pick up something and deposit it somewhere else. So I think it's been pretty well disproven with HIV and other blood-borne pathogens. So I think it's pretty unlikely that you'd be able to get spread. And if it was happening that way, the epidemiology would be completely different. Right? So,
well, it's very interesting when the public gets involved in this. They always come down to, well, how do you know? Because we say, as you said, that this would be different in this, but then, well, how do you know? And in science, you know what the data tell you, basically. So I think there have been a few studies where they have put Ebola virus into mosquitoes of different sorts and doesn't replicate. So in your view, that would most likely rule out mosquito transmission, right?
Yeah.
They've also sampled lots of arthropods in areas where there have been outbreaks, and they don't find the virus in any of them.
Right.
But I think your point is the most important, that the epidemiology would be way different, right? It wouldn't just be limited to the forest areas where the bats and the non-human primates are carrying virus, but other regions as well.
Right.
So in general, when mosquitoes pick up these viruses, yellow fever, chick, dengue, SINBIS, does it have any negative effect on the mosquito?
In most cases, it doesn't seem to, and that's kind of what got me interested in switching to that field, because I had been working in apoptosis as an antiviral defense in baculoviruses for a long time. And surveying the literature, you find these scattered reports here and there of some cytopathology in mosquitoes that are infected with a particular virus, which looked like it could have been apoptotic type of death in some cases. But I think by and large, when you look at a successful vector-virus relationship, which is what people naturally tend to look at, you don't see any kind of you don't see very much detrimental effect, if any, on the vector, and that's probably the reason why it's evolved to be a successful relationship. The virus is parasitizing the host without, or with the vector in this case, without damaging it enough to make the vector evolve a mechanism to get rid of it.
How long does a mosquito live, typically?
The Aedes aegypti mosquitoes we work with in the laboratory live for several weeks. We can keep them alive for 2 or 3 months. In the wild, obviously, it's probably much less because they're not being pampered like they are in the laboratory.
So when they're infected with these viruses, is it for the lifetime of the mosquito, or is it a very brief acute infection?
No, it's a persistent infection. They get infected. They seem to be pretty. Their high levels of virus can be detected for the rest of the life of the mosquito.
And the mosquito's fine as far as you can tell?
I mean, there have been studies, some studies have said that there's some negative effect on lifespan or fecundity. Other ones have said there isn't, so it's kind of, if there is an effect, it's a subtle one.
Yet, you said it's the only way that mosquitoes, and this is only female mosquitoes are infected in the wild with viruses, right? And only about 10% of any given population.
Or less.
So they do not spread it among them, obviously, right? Or am I assuming?
No, not directly.
It's just from biting hosts?
There is vertical transmission of some of the arboviruses from the female to the offspring.
Through the eggs,
Through the eggs, right? If the ovaries get infected, you can get, it's called transovarial transmission. But But no, horizontal transmission directly from mosquito to mosquito, I don't think that occurs. No.
Okay. So, as far as we know, then all these viruses that infect mosquitoes have evolved, probably this goes back millions and millions of years, they've evolved to be neutral. They're still replicating,
Right.
but they're not causing damage to the host. And it's not the case for a lot of human viruses. For some it is, but many human viruses cause damage. So what's the difference, do you think?
I think work of other people has shown pretty clearly that the immunity in the mosquito is suppressing the virus to the point where it's not pathogenic anymore to the virus. So the RNA interference seems to be the main defense in mosquitoes against the RNA viruses.
Okay.
And other people have shown that if you eliminate the RNAi response, the viruses become pathogenic to the mosquito. They kill the mosquitoes.
Okay.
So I think the mosquito has evolved to suppress this virus down to the point where it's not damaging the mosquito anymore. But there's no selective pressure to eliminate it because it's not damaging it anymore.
So this is the perfect arrangement, right?
For the virus, yeah.
And the host too. The host doesn't get sick. The virus gets to propagate. So why haven't we reached that point with polio, measles, mumps, rubella, smallpox? Why do we get sick?
Yeah, I don't know. It's a good question.
And why questions are not allowed, I know.
Yeah.
What's the mechanism that allows us to get sick? I mean, I guess we haven't been around long enough, right?
Yeah, I think evolutionary biologists would probably say that it's just a matter of time, right, until you evolve.
Yeah.
This arms race comes to a point where it's not pathogenic anymore.
I think that that's what I think, because most of these viruses have entered humans pretty recently. So we're not that old a species, so they haven't had much time to evolve. So Ebola is probably relatively recent, but maybe in thousands of years it would be much more mild. I don't think we've been studying viruses long enough to really know in people. It's only 100 years that we know about viruses, right? So I think it's a really interesting idea that if you look at other species, and there are others out there, other virus-host relationships that are benign also.
And a lot of times a human is sort of an accidental host, right? I mean, these viruses didn't really evolve to infect humans probably.
Yep, absolutely. So do the viruses give any benefit to the mosquito, you think?
Not that I'm aware of. No, not that I know of.
Because there's some thinking that maybe, we have lots of herpes viruses in us, that they may be beneficial. And in a mouse, you can show that a mouse herpes virus is beneficial when you infect them with bacteria. So I'm wondering has anyone looked at that?
Not that I know
that you know of, yeah. Because otherwise you might imagine that the mosquito would try and get rid of it, but maybe it's not possible.
Yeah, I mean, it wouldn't be surprising, I think, if there was some kind of benefit.
So you've done a lot of work on apoptosis in mosquitoes and with respect to virus infection. What's the connection there?
Typically, we think of it as being an antiviral type of response in mosquitoes or in other kinds of insects. And the work that we did in baculoviruses earlier, starting in Lois Miller's lab, showed that it is a pretty effective antiviral defense against those viruses. But baculoviruses, of course, are very different from they're large DNA viruses that need the host cell to stay alive for a certain amount of time so they can replicate their DNA and finish the replication cycle. So they, like a lot of large DNA viruses, encode genes that inhibit the apoptotic death to allow them to finish their replication.
In fact, the first anti-apoptotic proteins were discovered in baculovirus, isn't that correct?
Yeah, that's some of the first ones. That was work I did as a graduate student, yeah. So those were some of the first ones. I think the EBV, not EBV, the adenovirus E1B protein was known to have similar type of property too at that time.
So for the RNA viruses, though, it doesn't have a similar role, right?
Yeah, well, it seems to be. So I think it depends on the virus and the situation. Sometimes it's actually involved in the pathogenesis of the virus. So with certain RNA viruses that like most of the arboviruses actually induce apoptosis in mammalian cells, in neurons, for example, in the brain, and it contributes to the replication and the pathogenesis of the virus. But in the mosquito, those same arboviruses usually don't induce apoptosis. It's usually a persistent infection, non-lytic infection. So we got interested in, could this be a defense response in the mosquito? And maybe we just don't see it because, again, we're looking at an evolved, successful co-relationship between virus and vector. And if you look at, perhaps in non-successful, maybe one of the reasons why it's non-successful sometimes is due to something like apoptosis, a non-compatible response between the virus and the vector. So we tried a couple of different approaches to look at that. We wanted to test what would be the effect of, if we induced apoptosis, what would be the effect on the virus in the mosquito. So using Synbis virus and Aedes aegypti as a model system, we took two approaches. We first used RNAi to knock down genes in the mosquito that we knew would either positively or negatively regulate apoptosis. And we infected those mosquitoes then with the virus. And we found that when we knock down a gene that is required for cell viability, so if you knock it down, you cause spontaneous apoptosis in mosquito cells. That gene's called IAP1. When we knock that gene down, first of all, about 60% of the mosquitoes just keel over dead within a couple of days because you're inducing lots of apoptosis in the mosquito. But the ones that survived, when we infected them, they actually were more susceptible to infection with Synbis virus, which once we got that resolved, we started thinking about what we were doing and about how we were affecting the mosquitoes. And I think we were having pretty negative effects on the mosquito, probably the immune system of the mosquito. We knew that from looking at the guts of the mosquitoes were very altered, very fragile, and they didn't look normal at all. So by inducing apoptosis sort of globally in the mosquito, I think we were just weakening their responses. And so, but the interesting thing was when we knocked down a gene that was required for apoptosis, a caspase, we actually saw the opposite effect, that they did become a little bit more resistant to infection, which suggested that caspases were somehow involved in the replication of the virus. And that kind of led to collaboration with Lorena on the work that we're doing now, and when she tells you about what she's doing, that'll be more clear why. But then the second approach we used was to express genes in the virus that would cause apoptosis. So that would only be in the infected cells instead of globally.
Right.
And so we used a gene called Reaper, which is a gene from Drosophila. It's a small 65 amino acid protein that's very strongly induces apoptosis when it gets overexpressed in cells. And so we found that when we infected mosquitoes with the viruses expressing Reaper, there was a disadvantage for the virus. So in the initial stages of the infection, there was less of the mosquitoes got infected, and the titers were lower in the mosquitoes. So Reaper expression causing apoptosis did seem to confer some resistance to the mosquitoes. But the interesting thing was the infection caught up over time. So by about a week after infection, the titers were the same as the control viruses. The control virus we used had the same Reaper insert, but in the opposite orientation, so it wouldn't express a protein. So then we started sequencing viruses that were coming out of those mosquitoes, because with the thinking that perhaps there was selective pressure against retaining that Reaper insert in the mosquito. And sure enough, that's what we found, was that very rapidly after infection, the mosquitoes infected with the Reaper expressing virus, the Reaper insert was deleted or mutated, but not in the ones where the antisense was there. So it seems like there's very strong selective pressure against this induction of apoptosis. So I think it's pretty good evidence that apoptosis, if it does happen, it can be a very effective defense against the virus.
Okay, so in a natural infection, this has been modulated so that the virus is able to replicate.
That's what we hypothesize. Yeah, we don't know that, but
So you've collaborated to understand how this works? How he said a caspase is needed for replication.
Yeah.
Can you tell us more about that?
So our collaboration sort of happened by chance. Our paths basically crossed. So I was working with baculoviruses, and I started working. So baculoviruses are large DNA viruses. One of the amazing things about them is that they have so many genes, about 150 genes or so, that, I mean, they can basically play the host to their own will. Although I guess polioviruses do, and they barely have any genes.
I would say they have one gene, but they make 12 different proteins, yeah.
Yeah. So that's one of the exciting things about
some don't have any
with
genes. Viroids have no genes,
yeah,
right?
Yeah.
And they do amazing things.
Yeah. Yeah. So that's one of the exciting things about baculoviruses. I mean, they have so many genes. And we decided to study a gene that nobody had studied. And this was back when all the viruses were being sequenced. So it had been listed in the sequence that there was a gene that had homology to a fibroblast growth factor, FGF. And when you read the literature about FGFs, I mean, they are upregulated during cancer metastases in mammalians. They induce cell motility to differentiate certain cells, etc. And I couldn't understand why would a DNA virus encode FGF. When you turn to the literature of Drosophila, which also encodes an FGF, we found some surprising functions for FGF that could be tied into the baculovirus replication cycle. So in Drosophila, work done by Krasno, which is a Howard Hughes investigator at Stanford, what he had seen is that FGF in Drosophila basically defines the pattern, the branching of trachea. And trachea are basically a network or tubules, sort of like our veins, that basically deliver oxygen to every tissue in Drosophila. So he discovered, with very elegant tools, as you may in Drosophila, that when FGF was secreted from a cell, it was a signal that basically migrated to the tip of tracheoles, interacted with a receptor, and that tracheal cell could migrate towards the source of FGF. So there was cell motility, cell growth, not cell proliferation of the tracheal cell. So if you have FGF expressed here, that tracheal cell migrates to that source of FGF. And thus, depending where FGF is expressed, you start making that branching pattern of trachea. So the curious thing and the corollary to baculoviruses was that back in 1990s, in Lloyd Boltzmann at Berkeley, she sort of realized that so baculoviruses get taken by a Lepidopteran larvae through the mouth. They infect midgut epithelial cells in the gut. And like Rollie alluded to, it is not clear how they escape the midgut, because the midgut epithelium is lined with a basal lamina, and viruses cannot cross.
They have to cross them in order to reproduce or to spread to another animal? They
have to, well, not like in with salivary glands, but what happens is they escape the midgut, and then basically you can produce more virus in many, many tissues till the insect basically, we call it melting, because it just dissolves. And then the virus is released. And
it drips down through the trees.
Yes, exactly. Drips
onto the leaves.
Yeah, the virus sort of goes up to the trees, melts, spreads the virus.
So if the insect or caterpillar just ingested the virus and passed it through, this would not amplify it?
No, although there are some baculoviruses that are midgut restricted, and they kill the insect.
So they just replicate in the midgut, and then they're shed.
Yeah.
They're not shed, they kill, they melt the
they
insect?
kill, and eventually I think the insect sort of disintegrates and then the virus can get spread.
Okay.
So that has happened. So going back to FGF, so it turns out since the virus cannot escape the midgut, or eventually does, what Lloyd Boltzmann saw was that there were tracheal cells that sort of needed to oxygenate the epithelium of the midgut. So these tracheal cells sort of pushed through that basal lamina that lined the midgut, and they were infected. So her hypothesis was that the virus that, after infecting the midgut epithelium cells, used tracheal cells as a bridge to escape the midgut and then infect the other organs that are present in the hemocoel.
And that's because the virus is producing FGF that...
So that was our hypothesis. So our hypothesis was that if the virus expresses FGF, it would induce motility of the tracheal cell, infect that tracheal cell, and then spread. So the curious thing was that tracheal cells are epithelial cells. They have a tube which is made out of chitin where oxygen gets transported, but around that chitinous tubes there's an epithelium. So since there's an epithelium on the outer layer, there's also a basal lamina. So it wasn't clear why would the virus escape the midgut through the midgut epithelium versus that has a basal lamina, or try to infect the tracheal cells that also has a basal lamina. So to me, FGF was the answer to everything. So the way we pictured it, and we haven't proven it, is that when you induce cell motility, in order for that cell to grow, to expand, it needs to break that basal lamina. And in that window of time, when that tracheal cell has motility and it's breaking that basal lamina, then the opportunistic virus infects that epithelial cell of the tracheal cell and escapes. So the work that we did is, first we saw the corollary that these midgut-restricted baculoviruses do not encode FGF.
These are naturally occurring, right?
Yes.
No FGF.
And all the viruses that, as far as I can tell, that establish a systemic infection encode FGF. So that correlation basically made us hypothesize that FGF was required, or at least facilitated midgut escape, and possibly through the tracheal system. So the first thing we did was knock out FGF. When we did that, the virus still escaped the midgut, but much, much slower. There was a delay. So all that sort of seemed reasonable. But we did some electron microscopy, and we tried to look at the basal lamina. And the basal lamina of tracheal cells had become very mushy, almost like a little spongy, instead of being a thin, straight layer.
In animals infected
in animals
with
that
the deleted
infected, no, the ones that had FGF, the basal lamina was disrupted. If you lacked FGF, the basal lamina, it was changed a little bit, but it was basically like the normal basal lamina. So that was another clue that the basal lamina was being tampered in the presence of FGF, perhaps degraded. So what didn't make sense to us is usually basal lamina remodeling. All the proteins in the basal lamina are remodeled by matrix metalloproteases. So we started looking at the role of matrix metalloproteases in all this.
The virus encodes them.
Yeah. Most baculoviruses do not encode matrix metalloproteases. Some do, but we think that they have a different role. Which is another story, and we are studying those too. So we looked at matrix metalloproteases, and what we did is we used basically some mammalian antibodies to look at that. And we also looked at inhibitors of matrix metalloproteases. So what we did is we fed inhibitors of matrix metalloproteases to larvae and then infect them with virus. So when you block matrix metalloproteases, you don't mount a systemic infection.
Even with FGF?
Yeah. So we did a lot of in vitro and in vivo results, and what we found was that it seems that FGF was somehow, I don't want to use the word activated, but was sometimes cross-talking to MMPs, matrix metalloproteases.
So the midgut restricted baculoviruses don't have FGF, so they don't talk to the MMPs. That would be your hypothesis?
The MMPs that I'm talking about are host MMPs.
Right.
Host
Are they induced by FGF?
They are induced by FGF. They are activated. We saw that in vitro and in vivo. So they are activated by FGF, and they need to be activated. And so the curious thing was that the postdoc that was working on this, his name is John Means, he had seen one paper on the relationship between caspases and MMP. And he said, I'm going to use an antibody to a caspase from Drosophila and see whether caspases have any role. And I said, okay, it's a simple experiment. Go ahead and try it. So when FGF is present, you see caspase activation. And this is
where?
In the insects.
In the midgut
What he
cells.
did is he just basically laid out all the midgut that has a lot of tracheal cells on top of it. And with the antibody was FITC labeled, I mean, he could see it glowing. And in the absence of FGF, he didn't see caspase activation. So the viruses that had knocked out FGF, no caspase activation. If you fed the insects with an inhibitor to caspases, you don't see basal lamina degradation, and you don't see the virus escaping the midgut. So, a long story, if I can put a long story in one sentence, basically what we saw is that FGF somehow activated, and I don't know how many steps are in between, matrix metalloproteases. And matrix metalloproteases activated, and in vitro they do, and in vivo also they do because we play with inhibitors, caspases. And when you have caspase activation, then you have midgut escape.
So why, if you knock out FGF, is the virus still able to cross the midgut?
So the way I envision this is a basal lamina, either the basal lamina of tracheal cells or the basal lamina of the midgut, are in constant flux. There's a turnover of proteins all the time. And somehow the basal lamina creates little gaps that the virus might be able to escape through. So I think at some point the virus evolutionarily acquired FGF from the host, and that facilitated midgut escape. And some people have done some phylogeny work and evolutionary work, and the viruses that are midgut restricted are more ancient viruses. So those hadn't acquired FGF, and maybe acquiring FGF allowed the virus to basically express its host range, enabled to infect more insects.
That's how we're interpreting it, right?
Yeah.
Who knows?
That's how we interpret it.
I always am suspicious of things that we think up that we believe will help a virus,
how we justify
right?
them, yeah.
Because we don't actually know what the pressures or the selections were, right, at the time. And it may have been very different back then when it first acquired the FGF than it is now also, right? Because there's this, evolution doesn't have this long-term view. I'm going to be really good in a million years, right? It's right now. So if you take a virus lacking FGF, there's no caspase activation, is that correct?
Very low.
Okay,
Very
there's
low.
some.
So that background level might allow the virus
To get through. Right.
Even in the
Got
absence
it.
of FGF. So the way the midgut works, it's an arms race basically. So cells in the midgut get infected, which is the primary site of infection, and they get sloughed off
Right.
So if they get sloughed off,
normally they're sloughed off just like ours, right?
yes,
Yeah.
all the time. So, but if there's a virus infecting those midgut cells and they get sloughed off, you lose infection.
Yeah.
So I think the virus doesn't have much time to really escape
Right.
the midgut before the cells get sloughed off. So acquiring FGF, I think, is a time thing. They can quickly escape the midgut and establish the systemic infection.
So have you ever thought of putting FGF in one of your non-mosquito infecting viruses?
Yeah, so actually, this is what led to the whole collaboration.
Yes.
We started wondering whether similar things were happening with arboviruses in mosquitoes, whether they were also using an FGF type pathway. Arboviruses don't express FGF naturally, but they are also blocked by that basal lamina from escaping from the midgut. So we have actually tried to express the baculovirus FGF in Sindus virus. This was work done by a postdoc in Lorena's lab, Wenbi Wu. Didn't really see much of an effect, though. It didn't seem to accelerate.
But the FGF was made, right?
Yeah.
It was made, yeah.
It's synthesized. But
And you didn't see caspase activation?
There was some caspase activation, but not as much as we thought there would be.
Okay.
Yeah.
So putting
and the background is fairly high too, so it's difficult to interpret whether that makes a difference or not.
So putting in FGF into Sindus is not enough to have it cross.
No.
it didn't seem to accelerate it
No.
or stimulate it.
But this isn't a mosquito where it would cross anyway, right?
Yeah. So we were using a virus, a strain of Sindus virus called the TE strain, which does not naturally infect very well in the mosquito. It's a tissue culture adapted strain. So it has a difficult time establishing infection in the midgut and escaping out of the midgut. So we hoped that would perhaps allow it to give it an advantage, but we didn't see that. But we're still investigating that pathway and how it affects, if it has any effect on virus escape. So we've looked at MMPs, we've looked at the endogenous mosquito FGF. We're still looking at that, and caspases, some of the caspases as well, to see if when you either knock down their expression or overexpress them, that influences virus replication.
Because your original observation was that caspase activation seemed to be required for efficient infection in mosquitoes.
Right. So that's what
and
kind
then
of
the
led us to
baculovirus has provided one idea why this might be through FGF activating, and you're trying to now see if that applies to mosquitoes. Would it be worth taking a mosquito that's normally, that Sindus doesn't cross the gut and seeing if, because maybe your mutant has some other problem that is preventing FGF from working, right?
That's certainly possible. It doesn't establish infection very well in the midgut either.
Are there such mosquitoes that would be refractory to the virus?
Yeah, there are certainly some.
But probably you don't want to work with them because you have your standard
we haven't gone into
colony.
that, but it would be a good thing to try.
Can you tell us for the listeners, what is a matrix metalloproteinase?
Well, usually these proteins, what they do, they're secreted, they're present in the matrix. And for example, the basal lamina is composed of, it's basically proteins, it's acellular, and it's composed of proteins like laminin, nitrogen, and they basically remodel them, which means they chew them up, they cut them, and then they get resynthesized.
Okay.
And what the basal lamina really does, one of the things, I mean, it's a barrier, but it also developmentally helps cells sort of have a framework in order for them to sort of replicate along a line so they can form an epithelium.
Okay. And the metallo simply means there's a metal cofactor needed for that, right? Okay. So can we go back to baculo and talk a little bit about the general biology of these viruses? So these infect mainly caterpillars or other?
Yes, and most of the viruses that have been studied and isolated from infect Lepidoptera insects. The larval stage, which is just a little caterpillar.
Right.
They don't infect the adult, which are moths. They have an amazing replication cycle. So the larvae will consume foliage, and the foliage will be contaminated by one form of the virus and gets taken into the midgut. So the form of the virus that gets taken into the midgut, the virions are protected by this matrix, which is made of one protein called polyhedron that many people know because that's the one that you substitute to have foreign gene expression. So this polyhedron, this matrix, gets basically dissolved in the midgut of an insect, which has very alkaline pH, like pH 11. So once you dissolve this matrix, this polyhedron, the virions can attach the microvillus of the epithelial cell, enter the epithelial cell, and replicate.
Right. And hopefully cross.
Hopefully cross. There's a hypothesis that some viruses actually replicate, but the fusion protein can be made early and late. So it can be made early so it can get to the basal side of the epithelial cell. So some virions just don't replicate. They just basically just go through acquiring a fusion protein and exit the midgut quickly in case those cells get sloughed off.
Okay.
So the virus will replicate, will produce budded virus, and the budded form of the virus is basically acquires the membrane when it comes out of the cell.
So you have two forms. You have a budded,
yes, the budded virus and the occlusion virus.
That's with the polyhedron.
That's the one that is in the.
Why is there both?
So the budded virus allows spread between cells.
Within the insect.
Within the insect itself.
So that doesn't get outside.
Well, if it gets outside, it'll get just desiccated.
Degraded. Okay.
And the occluded form of the virus, when the insect melts and it rains its virus, it's protected. It's very, very stable in the environment. It can stay in the soil for a long time. It gets damaged by UV light, but other than that, I mean, it's very stable, and that's the form that it gets consumed.
Okay.
If you take a budded virus and you feed it to an insect, I mean, the alkaline pH
will destroy
it. Allow it. And it also so the proteins in both forms of virions are different. One is necessary to infect between cells to cell, and the other proteins are necessary to infect the midgut cells.
Now, I understand that if I buy cabbage and eat it, it's covered with baculoviruses.
Yes, you have it.
So what happens when I eat these baculoviruses and they go into my acid stomach? Nothing happens?
No.
They don't degrade, or they just pass through?
They just pass through.
Yeah, they might be degraded, I think, probably.
By the
acid. They could be
acid.
degraded in the acidic.
So even with the polyhedron, it'll be degraded and would never infect us, right?
No. No, actually, long time ago, I read a review somewhere in there, and I couldn't even give you the reference. But apparently, back when they did some experiments, and then they actually fed people globs of baculoviruses, and people were fine.
Nothing happened.
Nothing happened.
But but the budded form of the virus can enter mammalian cells pretty efficiently, actually. So that's one of the things that the viruses are being developed as actually gene therapy vectors, because they can transduce human cells. They can enter, uncoat their DNA, but the genes of the virus aren't getting expressed because the promoters aren't right for to be expressed in a mammalian cell, at least most of them. But so they don't replicate, but if you engineer the virus to have a mammalian promoter, you can drive expression of foreign genes.
And it would be very safe as gene therapy vectors because they don't infect.
They don't replicate.
Right, they don't integrate. They're not going to integrate, so that's not a problem. So this is actually being developed now for people. What kind of diseases do you know? Just monogenetic diseases?
Yeah, I mean, I think basic research just says trying to develop them as gene therapy vectors.
Do you think people would have a problem getting an insect virus for therapy? You know, people have funny views about
insects.
It should be less of a problem than getting an adenovirus for gene therapy or a retrovirus, I think.
In theory, yeah. So baculoviruses have been used for many years to express genes, right, in insect cells. But this is a different application. This is actually to make particles that can infect.
Yeah.
How do the viruses infect human cells? Do we know?
They think some electrostatic interactions.
There's not a specific receptor.
We don't know about a receptor. We think it's a very common receptor because it enters a lot of cells. So it must be a very common receptor. In the laboratory, you need a lot of virus, high multiplicity of infection in order to infect those cells.
The human cells.
But human cells.
Would it help to put like a VSV G protein in and then get really efficient infection?
That's been done. Yeah.
It works?
Yeah. But they're pretty efficient, actually, on their own. Yeah.
So you would inject this into people and supply a gene, or maybe take cells out and infect them in culture stem cells and put them back in?
That would be the
that's
idea,
interesting. So, all right, so back to the caterpillars. So they're eating the leaf and they ingest the virus, and as we've said, it can replicate in the gut, perhaps traverse the gut cell and spread. And you say the caterpillars liquefy, and then the virus drips down onto the leaves, and that's how it gets to other caterpillars, right? And I read somewhere there's a protein that the virus produces that makes the caterpillars crawl higher in the trees, in the plants.
Right.
Is that true? Can you tell us about that?
EGT.
One of your papers, I think. Yeah.
No, I didn't do that study, but there was a review on it. Yeah.
Maybe it was in a review. Yes, that you wrote. That's right.
EGT was an old study done by David O'Reilly. So let me see how it goes. So EGT basically transfers the glucose to ecdysteroids, which are molting hormones. And so the virus encodes this gene to prevent molting of the insect. So
molting
we need prevent, yes.
Becoming a moth, right?
Yes.
Okay.
Yeah, becoming a pupa first, or molting. During molting, the larvae basically are inert to some extent. So.
And the virus doesn't like that, right?
Yes. Yeah. So the virus kept the caterpillar going.
Right.
But with you delete the gene, then the insect molts.
Right.
So they patent this as a pest control agent because since the insect stops eating because it molts.
Got it.
It stops destroying, consuming crops.
But the virus, from the virus's perspective, this is good because it allows replication longer.
Yes,
right?
yes.
And the caterpillars get bigger. And then they burst,
the virus
right?
gets
lots of
more
virus.
Yes.
So what I was thinking.
EGT basically, and that was some work by Kelly Hoover. So she's actually saw that it was responsible for going up.
Right. So what I was thinking of is this review that you two wrote in PLOS Pathogens. There's this phenomenon called Whipfeldkrankheit.
Yeah.
Can you say it?
No.
It's a German word.
Whipfeldkrankheit, the baculovirus-induced climbing behavior.
Right.
What's that?
So that's when they
start to molt, I
think there's another gene also, there's a protein, tyrosine phosphatase, that's encoded by the virus, and that also, both of those genes have activity of basically modifying the behavior of the caterpillar, causing it to seek out, go up the plant, and then, which normally they don't do because it makes them more susceptible to predation and things like that.
Right.
And then they melt. So, and the melting actually is interesting too because there are two viral proteins that the virus expresses which actually causes this melting of the caterpillar, disintegration of the caterpillar. One is a chitinase enzyme, and the other one is a protease called cathepsin. And so both of those enzymes together sort of liquefy the caterpillar and cause it to basically become a puddle of slime that's full of virus.
A lot high levels of
very
virus,
high levels of
10
virus.
to the 9th.
Amazing.
Yeah.
Total particles per caterpillar.
Right.
And the higher you go in the plant, then it can spread on more leaves. That's how we're looking at it.
Yeah.
Right?
Yes.
Yeah.
And other caterpillars will eat it and it will spread, right? So this is a virus that has an effect on its insect host.
Yeah.
Kills it.
In most cases, yeah, very pathogenic.
As opposed to the mosquito-born viruses
Right.
that we talked about, which are persistent infections.
So a different life strategy, yeah.
But both work.
Yep.
So our view, you know, we have this human view that all viruses should evolve to be persistent and harmless, but baculos have not.
Baculoviruses burn their bridges
They
as
burn.
they go.
I guess as long as there are enough hosts out there, it works, right?
Exactly, yeah. And they can, like Lorena said, they can survive in the soil in between these
Yeah.
epidemics
Cycles.
too. So although they do in the wild, there are cycles of, and it, you know, tracks along with the caterpillar population cycles as well. So as the caterpillar population increases, then you're going to probably more likely to get an epidemic of virus that brings it back down, etc.
So if you go out and sample populations, you get what percent are baculovirus positive?
I don't know about percentage, but
10% like the mosquitoes and their viruses?
Yeah, I don't know the numbers,
numbers.
but,
No,
but
you
it's
would
pretty
find
common
that.
to find it.
If you went out in a field, you would find it in any
Especially in the fall, if you go for a walk in the woods, you probably will find caterpillars hanging down from a tree, and they look black, and they're
Yeah, I've seen
kind
those.
of disintegrating. I mean, those are virus-infected caterpillars.
Yeah. And this can be used to control pests?
Yeah, so
It is
they
being used.
It is being
they have
used,
been
yeah.
used a lot in the past. They still are being used as control agents. Yeah, they have a lot of advantages because they're very specific for just they only have certain hosts that they infect. So has no impact on other non-target insects, beneficial insects.
Brazil is a big user. China too.
How would they be applied to plants? Would they be sprayed?
Yeah, they are sprayed. Yeah.
So they're a
Even
liquid.
from a plane,
Oh, so the
Yeah.
liquid formulation, and the plane goes and spray, and because
and it's the occluded form of the virus.
so it's very stable. So there are companies that produce gallons and gallons of this, right?
Yeah,
Yeah.
and you
So-
can buy them in organic farming. You can get it. You know, if you're a small organic farmer, you can
Yeah.
buy these products as well. The disadvantage is that they're too specific, right, in many cases. So,
host specific, you mean?
yeah, so depending on what type of pest you have in your garden, you need to find the virus that can kill that particular pest.
I see.
If
So
you
also-
have-
yeah.
and if,
How would you do that? How would you determine? How would a farmer determine which? By looking at the species of caterpillars?
identifying
Yeah,
the species
yeah,
and
yeah.
then seeing if there's a
And the
product that
pathology,
can
yeah.
that can.
But if
Okay.
you're a farmer,
Affect it.
you know, and you could use a chemical pesticide,
It's easier, right?
it's easier, you know.
Easier,
you just,
cheaper,
yeah,
faster.
easier and cheaper.
But it's
than trying to-
toxic to us when we
Exactly.
eat it, right?
Yeah.
And it
So-
runs off and it kills other things too. So that's why this is organic, right?
Right.
Because it's not a chemical and it's biological.
Right.
But it's harder to do. And it's interesting. Well, there are other viruses too that can be used as
Yeah.
pest control, but they probably have similar limitations that you have to figure out. You know, this reminds me of using phages to treat human bacterial infections, right? This is being worked on for many years. The problem is you have to identify the bacteria and find a phage that would be
Very
specific.
specific.
Okay.
So you have to tailor make the drug each time someone has an infection. So that's, I think, a limitation to that. It's a similar idea as well. So what else about baculos is fascinating that we haven't talked about? Do you say, so these viruses replicate in the nucleus, right?
Yes.
And so the DNA is replicated there. Where are the particles assembled? Also in the nucleus?
In the nucleus.
And then how do they get out of the nucleus?
We don't know how they get out of the nucleus. They get out of the nucleus.
As polyhedra-covered
The
particles?
bug-
No. So at late times,
Right.
the nucleocapsids that are produced in the nucleus come out of the nucleus, transit through the cytoplasm, and brought out of the cell. But at very late times, and we don't know what makes the switch, or it's not clear what makes. Then they stay in the nucleus, and then they get occluded with polyhedra.
Okay. So then we don't know how they get out. Somehow the cells break open and
That's how the
Yeah.
inclusion
Particles.
bodies get out. But
Yeah.
the budded viruses
They
just
bud out,
bud
sure.
out like
Yeah.
a normal
so a cell making budded viruses will do that for a while,
couple of days, 2 or 3 days.
days,
typically.
and then it becomes full of occluded particles.
Right.
And at some point they'll break
The
open.
cell lyses eventually. Yeah.
And is that lysis because of apoptosis, or we don't really know?
It seems to be just sort of passive necrosis type
Okay.
cell death at that point. Because
So-
the virus is actively suppressing apoptosis during the replication.
And that's done because it wants a long time to replicate.
Right.
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 What is this, the replication cycle? How many hours does it take from infection to getting first infectious particles? How long does it take?
Well, in cell culture, it's 48 hours.
Okay, so it's pretty long.
Yeah, yeah. I mean, to kill an insect, depending on the virus, anywhere about 5 days, say.
Okay.
But there is virus produced starting within 12 hours in tissue culture.
I mean, the budded virus, yeah.
Budded virus,
Yeah.
And for comparison, polio, within 4 hours you get particles made. It's almost like a phage.
Yeah.
It's so fast.
Sindus virus. So
Fast too, as well.
Fast, much faster. Yeah.
So let's, to end up, let's go back to mosquito-borne viruses. And I wanted to ask, well, both of you, do you view some of this work as contributing towards control of mosquito-borne diseases in any way, or is it really fundamental basic research that that involves unraveling mechanisms?
Yeah, I mean, mostly what we're doing is fundamental things, just trying to understand what it is. But I think, the hope would be that if we understand what makes some mosquitoes resistant and other ones not, then we could somehow try to tailor that into some kind of approach to genetically engineer mosquitoes. I mean, there are people that are working on that, trying to do that, and with the hope of one day being able to release those mosquitoes that are now resistant to virus or malaria infection into the wild and be able to curtail some of these epidemics.
So whatever you did would have to not exert any fitness costs.
Yeah, that's the problem, right? Is whenever you start tinkering with the genome, you're probably going to make it worse.
Always.
Yeah.
Same with viruses.
But there are driver mechanisms that people are trying to develop to make, to try to drive these genes into the population.
So I'm sure you know about the Wolbachia experiment where they put Wolbachia into mosquitoes. It makes them refractory to dengue, I think, in particular.
Dengue,
and do those have a cost in the mosquito?
It doesn't seem to be, no. I mean, the majority of insect species on the planet already are infected with Wolbachia, which is an intracellular bacterium or some endosymbiotic bacterium.
So the mosquitoes you have in your lab, they don't have Wolbachia?
No, Aedes aegypti isn't naturally infected with Wolbachia, but what researchers have done is find Wolbachia strains and artificially infect Aedes aegypti, and then some of those Wolbachia strains do confer resistance, pretty strong resistance to dengue. And
how about other arboviruses? Are they?
That's probably been looked at, but I'm not sure.
So do Culex mosquitoes have Wolbachia in them?
I don't know that either. Good question. But in places like Australia, Thailand, Brazil, they've released those Wolbachia infected Aedes mosquitoes, and it seems to be pretty effective at reducing disease transmission in that local area. They have to release a lot of them.
Yeah.
And the hope would be that they would be able to establish, the Wolbachia infection would be able to establish itself, because it does naturally get spread to the offspring. So if they mate with the uninfected mosquitoes, the offspring would be infected.
Right. So that's going to be a while before we can modify mosquitoes. In the meantime, what's the best way to deal with mosquito-borne virus infections?
Well, mosquito control is the #1 way. So if you can prevent people from getting bitten by mosquitoes, then you can prevent. That's the best way to prevent it. And with dengue, a lot of that involves just controlling the breeding sites of the mosquito, because Aedes aegypti has evolved to cohabitate with humans. It's in very close association with humans, and so the water containers and spare old tires and things like that that contain breeding sites are the #1 problem, because the mosquitoes don't move very far from where they hatch. Their range is only hundreds of meters. So by just eliminating breeding sites around people's homes, you can do a lot to reduce the amount of transmission. There are some work on vaccines going on.
I was going to ask you,
yeah, but then I think there's some promise with some vaccines coming.
So do we know since these viruses have to be maintained in people for the most part, right?
Dengue.
Except West Nile.
Yeah.
If you had a what fraction of immune population would it take to get now the likelihood that you're going to transmit by mosquitoes so low that it doesn't happen anymore? Do we have any idea?
Yeah, I think there are ideas. I'm not very familiar with that literature, but I've heard talks where people have talked about there is definitely a a theoretical threshold that if you can drive it down below a certain amount, then you would basically interrupt the transmission.
Right.
And
So
stop
you could eradicate dengue, for example, by immunizing a good fraction of people.
Yeah,
in theory.
it seems
Chick, you could as well, right? Because there's not an animal.
as far as I know, yeah.
But not West Nile because the birds
that's-
can't immunize birds.
Hundreds of species of birds that are infected, yeah.
Well, I was in Australia this year, and they have an interesting vaccine approach where to prevent Hendra infection, which goes from bats to racehorses to people, they immunize the racehorses because there aren't so many of them,
Yeah.
and that protects all the people and the horses as well. But that's possible because there are limited numbers of racehorses, and normally it's hard to immunize wildlife and other animals. I think that's a cool way to look at it. All right, thanks guys for doing that. I appreciate it.
Thank you.
This episode of TWiV will be at twiv.tv. You can also find it on iTunes and any podcatcher app. You can find it as well. You can find it on Stitcher and all the other places that podcasts can be found. And if you like what we do, best thing you can do to help us right now is to go to iTunes and subscribe and leave a comment there and rate us in stars. And that keeps us visible in iTunes and lets more people find us as well. And usually we answer questions on the show. If you have them, we'd love to get them. Send them to twiv@twiv.tv. So both of my guests today are here at Kansas State University in the Division of Biology, Rollie and Lorena Passarelli. Thank you both for joining me. Pleasure.
Thank you, Vincent.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.